Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this session to discuss NCERT Class 12 Chapter 4 Principles of Inheritance and Variations Part 4 Law of Segregation. This law is based on the fact that the alleles do not show any blending and both the characters are recovered in the F2 generation through the next to the law of segregation. This law is based on the fact of that alleles do not show blending and both characters are recovered such as the F2 generation through the one this also not in the F1 stage. See, so when you observe as in that both heterozygous characters, the capital T and small t, it is this F1 generation, am I right? It is not there blending these characters, but in this characters are not blending in that F2 generation. In that completely in that F2 generation, the characters are there completely. What happened? They get separated. Okay, that is called as what? That completely in a law of segregation. See that through the in a parents that contain two alleles during the gamete formation. So through the in a two parents they get complete their forming to as in a that gamete formation. The factors or alleles, the pairs are segregated of each another. Such gametes recessive, they complete the receive only for the in a two factors. The most they uptake as in a two factors. Okay, that is. Of course, the homozygous parents produce as in all gametes similar, while the heterozygous one produce two kinds of the gametes having the one allele with equal proportion. See, when you take as in the two gametes, it is the complete ratio as in a the equal proportion. Okay, this is next. Here, what is that? Incomplete dominance. Here, incomplete dominance. In that incomplete dominance, when the experiments on the pea plants where they are completely the repeated using as the traits of the plants, it was the found that in sometimes the F1 had is the phenotypic did not resemble to either the two parents was there having the two characters. See, actually in this incomplete dominance, the first there discovered by the scientist was in a corn corin. Corn corin in the plant is that is a in a 4 o'clock plant. 4 o'clock plant, that is nothing but as in a snapdragon plant. That is in a snapdragon plant. He is saying about is what the F2 the when the completely the F1 generation when you cross with as in a the characters, when you cross with as in a characters, here we getting as in that F1 generation. In this F1 generation, the heterozygous characters does not they are completely blending. Here they are not blending in that in a both homozygous parents. It is called as what? Incomplete dominance. This in the completely the flower cover they given, that is in a dark flower, that is a snapdragon or antherinum species. That is a good example to understand the incomplete dominance. So it is a best example for the good understanding purpose. And see they given here. One is that in a complete dominant characters, that is a red, capital R. This is in a red and red. That is a true breeding white flowers. The plants are smaller. So here this is smaller. It is in a white flowers. When that in a F1 generation, that we getting as what? Capital R and smaller. We getting as what? Here capital or smaller. This is F1 generation. Am I right? It is in a F1 progeny. Let us in that F1 generation, we getting as what? The pink flowers are noticed. We getting as what? Pink flower. When the F1 the self-pollinated, the F2 generation results the formation of the in a complete red flowers, and one is red flower and two is in a pink flowers and one is in a white flower, you notice. So when you observe here clearly, that is in a capital or capital red, capital or smaller in a pink color and smaller, it is in what? This is in a white color. Am I right? Here the genotypic the ratio when exactly as in that would be expected in that in a Mendelian monohybrid class. Means already when you observe here, it is a dominant character is a one only present. Here the heterozygous character is two is present, and then homozygous recessive plant is only one is present. So when you observe as in the both phenotypic, here the both here the phenotype as well as in a the genotype, the both ratio is to complete ratio as what? In a 1:2:1. The phenotypic ratios had the changed from as in a 3:1 the dominant the recessive ratios. Okay, what happened to be the capital R was not completely the dominant as in the smaller, while they made up of possible to as in a, that simply as in a capital R smaller, the pink and they show as in a red as well as in a white flower. So like as in a red, it is in a pink, it is in a white. Okay, next, what is exactly the definition of dominance? Okay, what is dominance? Explanation of the in a concept of the dominance. See, what is exactly dominance? Here, why the in some alleles the dominant and some alleles are the recessive? When you see here, some characters are the dominant, some are their recessive. Why? See, to tackle this the question, the must be understand what the genes does. Every gene here, you know that now the contain the information to express the particular trait, that is the diploid organisms. So when you observe as in the diploid organisms, here what happened? Here some characters they are completely expressed, some characters are there completely recessive. Okay, these are the two copies of the each gene. They are pair of the alleles. Now these two alleles they need for as any not only the identical in a heterozygote. So when you take as in the diploid organism, when one characteristic takes place here, so sometimes it is completely the dominant, but sometimes it is in a complete recessive. Means they show as in a homozygosity, and sometimes they show as in a heterozygote. So like means they're identical or not. Here one of them they may be they are different due to as in the changes that has in the undergo. Here when the about that which has in a read further one of the and the next chapter which is a modification the information the particular alleles they are contained. So this all information means the homozygosity when you observe as in that both parents they show as in different characters only. Here the only they contain as in a particular information or particular genes or particular alleles they get completely produced. Okay, that is example of the gene that contain as the information they producing the enzyme. So here then you saying about one particular that is the dominance. Here how do they express it? The dominant is expressed about as in only one condition, that is the enzyme is present. Now they are the two copies genes the two alleles form. So here two copies to get produced as in a two genes. Let us assume the more than common, that is the normal allele produced or normal enzyme to need for as in the transformation of in a substrate. Yes. So when you take as in this is in a like enzyme to get forms as in a substrate. Okay, like the theoretical the modification of the allele could be the responsible for the in a production of one is in a normal enzyme or less efficient of the enzyme, and next one is non-functional enzyme, and third one is what, it is in a no enzyme at all. So these are the complete that depends upon the it is complete character that is the dominant as in a recessive character. Okay, this is in a exactly the dominance. And after here the first case the modification of the allele equivalent to as any their unmodification allele. So in that first case of the modification alleles equivalent to as any unmodified. So here the capital D is there and small t is there. So when you see as in the first one and second one they completely show as any variations. Am I right? Here what will they produce the same phenotypic rates? The result the transformation substrate, that is a yes. Here the transformation is what? That is in a completely yes. So when you are in that enzyme active site to get bind with as in substrate, that is equivalently the allele the pace very common. So when you observe this entire characters, we see that in a very common. But alleles are produced as a non-functional enzyme, no enzyme. Here the phenotype may be there affected. So if in case the enzyme is present but there is a non-functional and no enzyme, but here what happened, that is also the completely affected as in that phenotypic ratio, means the physical appearance means the dominant characters will be they get expressed. Okay, and the phenotypic traits only when dependence upon the functioning the unmodification alleles. Here the unmodified also their functioning alleles which represent as in a original phenotype the dominant allele, the modified allele is in a the completely generally recessive allele. Which one they are modified, that is the dominant, which one is in the suppressive, that is completely called as recessive allele. Hence this in the example above the recessive traits the seen in due to as a non-functional enzyme because there no enzyme is produced. So when the completely enzyme is not produced, what happens, that characters will be the completely they are recessive. Okay, this is a completely incomplete dominance. It is complete what? Incomplete dominance. Let us we discuss that is a co-dominance. What is the concept of the codominance? Here codominance means, here when you see as any the heterozygous parents, here heterozygous parents, here we are completely, when you take as in that both homozygous characters, here homozygous characters we get cross self pollinated, we are getting as what? The F1 generation we are getting. So these both parents, the heterozygous characters, they are resembled to as in both homozygous parents, is called as what? That is in a process of the in a codominance. Okay, now let us, till now the we are discussing the cross where the F1 resembles either of the two parents, one is the dominant or it was between the incomplete dominance. But in the case of the codominance, the F1 generation resembles. See here, the F1 generation resembled to as in both parents. So these characters, for example, the tall is the tall, it is the dwarf, it is the completely dwarf. So the both characters resemble to in the F1 generation. That is called completely what? In codominance. Here they given as a good example for the different types of the red blood cells determined as an ABO blood grouping in the human being. So it is an ABO blood group. Okay, the ABO blood groups are then controlled by the which gene? That is in I gene. They are controlled. I gene control. The plasma membrane of the red blood cells has a sugar polymers that in a produced form as in surface. It kind of the sugar controlled by as in a gene. So in this, for example, you are taking as an I gene. The I gene controlled by as in a, they're completely the different kinds of their genes are there protected. Next, I gene has in the three alleles, that is in a IA, IB, and they're completely I. And the alleles are IA, IB, and they're slightly different as any, that's sugar, while the allele I does not producing as any sugar. So I is not produced as any their sugar, because the human diploid organism, each person possesses as any two types of the three genes alleles as present. Okay, any human gene they having as any their IA, IB, these two alleles as present in the blood group. Okay, next what is that here? IA, IB, here IA, IB are completely the dominant over I. So here IA and then IB, these are complete to dominant as what? In the I gene. So in other words, the IA and I, they represent as an I expressed, because their I is not produced as any sugars, and IB, I they are present B expressed, and but IA, IB, the presence of the together expressed as a type of the sugar. That is because the dominant is with the takes place. So IA, IB is forms, that one only. And hence the red blood group has in that A and B types of the sugars, and the different types of alleles. There are in a 6 different types of combinations. We see that in a possible for the in a total 6 different types of genotypic human being blood groups types as they are produced. Let us see in the table also. That is, here there is an allele there from parent 1, it is an allele parent 2, it is a genotypic offspring, it is in a blood type of offspring. See, IA is in a parent 1 and IA. So I genes are what? IA. So which blood group we are getting? A blood group is getting. And IB, IA, IB, these are the parent 1 IA contain, and IB they contain parent 2. So the genes genotypics comes as what? AB. So the blood group is what? AB blood group they are coming. Here next, IE, the IA they are completely, they have I does not contain any sugars. So the A is the dominant, so getting as in A blood group. And here B blood group is present, and A blood, here IB, IA. So these both are IA, IB, the genotypes we getting as in a AB blood group. And here IB is present. So here dominant character is what? That is a big. So in produced as an AB blood group. And IB, I gene does not contain any sugars. So getting as what? Completely in the B blood group. And antigens are the complete absent now. So they get produced as what? It in a O blood group is producing. Okay, this is in a co-dominance. It is completely co-dominance. Let us here after, do you as in realize that what the examples of the ABO blood groups, like providing the good example, they are multiple alleles. What they given, that is also one of the best examples for the multiple alleles. And you are seeing that in here the more than 1-2 genes or more than 2 alleles is present. That is complete if we get show as a morphisms. Like the three alleles group, they grouping the same characters. Since the individual only two alleles can be represent, multiple can be there found. Only the population study are made up of. So they completely are made up of what different characters they are forming. Here, occasionally the single genes produced by may produce more than one effect. For example, the starch synthesis, the seeds, they are controlled by one gene. So here they completely the two seeds we are taking, that is in starch synthesis seed. See here the two alleles, one is capital B and another small b. Here starch synthesizing affecting by any the capital B is the homozygosity, and their large starch grains the produce the construct that is the homozygous small b having a less efficient starch synthesis produced by any small their starch grain. It is in a more efficiency is present. Here is as in a completely their less efficiency is present. So after maturation, in that seeds of the capital B seeds and round and small b they are wrinkled. So it is in a completely show as in a round character. It shows as in what? It shows as in a that is in a wrinkled character. Correct. So when you cross here, we're getting as what the heterozygous character will be there getting. Right? The heterozygous they produce in a round seeds capital B and the same time the dominant allele, but the starch grains produced as in the intermediate that is in a size of in a capital B and then small b. So when you cross, what happened? It getting as in the capital B and then small b. So the starch grains they are considered as it is in a phenotypic. The from as in the angle allele it shows as in a incomplete dominance. So they show as what? It shows as in a completely incomplete dominance. It shows as in a incomplete dominance. And here, therefore, the dominance is not any autonomous the future of the produce that information for that depends upon the more genes the production of the particular phenotype as in it to form as in the particular phenotype. So here, once the genotype is produced, what the genotype produces in a particular phenotype as well as in a genotypic characters that we choose as in the examine the case more than one phenotype the influenced by the same gene. So the one gene to get completely they're influenced by as in a they're completely other same gene. Okay, this is the completely what codominance and information. Okay, remaining to discuss in next class. Thank you for watching. Like, share and subscribe.